This is the Biology Unit 5 revision video looking at excretion, the organs involved, the process and dialysis. So one of the main points that you need to know about excretion is that it is not the same as egestion. Now, excretion is where the waste is made inside your body, inside the tissues, and is then actively removed, whereas egestion, the waste is never actually made in your body, it just makes its way through your body via the tunnel, the tube that is your digestive system. Your body takes what it needs from the food and gets rid of the waste that it doesn't. The reason that excretion is so important is because a lot of the chemical reactions that happen in your cells make toxic chemicals, and if they're not removed then they can damage or kill the organism. Now, the main organs of excretion are the skin, the lungs, the kidneys, and also the liver is involved as well. You need to know what the main substances are that are excreted by these organs. So for the skin, the substances that are excreted are salt and water. For the lungs, it is carbon dioxide. The main excretory substances of the kidneys are glucose, salts, water and urea. And lastly, the liver doesn't actually excrete any substances, however it's involved in the breaking down of amino acids. So one of the reasons that you sweat is to cool you down, and water and salt from the sweat gland will travel via the sweat duct to the skin. Here the water in the sweat will evaporate, taking away excess heat, and will cool the person down. Now, it also works to get rid of excess water and salt. So if there is more water and salt within your body, this is one way that it can get rid of it. Cells in the body that are respiring will make carbon dioxide. Now, this is carried to the lungs here via the blood, diffuses from the blood into the alveoli and is breathed outside of the body. This means that your lungs excrete carbon dioxide. Now, the reason that they do this is because carbon dioxide is acidic and it will dissolve in the blood and lower the pH of the blood. Now, a lot of the chemical reactions that happen within the body rely on enzymes, and if the blood is acidic, then this can denature enzymes and could kill you. Your body will prevent this by the brain will detect the increased levels of carbon dioxide in the blood, and the brain will increase the breathing rate to remove carbon dioxide more quickly. Now, this is what the liver does in relation to excretion. As I said before, it doesn't actually get rid of anything from the body, but it's involved within that process. Now, the liver has many functions. One of them is to break down old red blood cells, and those chemicals, it will produce a chemical called bilirubin. They will go into bile and pass out with the faeces, and some of those chemicals give the faeces their brown colour. It also breaks down hormones and medicines such as paracetamol or other drugs such as alcohol. But here that you need to know in more detail, if you eat more protein than is needed in your body, then the liver will break down the excess amino acids. It changes them firstly into ammonia. Now, this is extremely poisonous to us. The reason being is that ammonia increases the pH, and enzymes will denature, they won't work properly, and this can kill you. Now, to prevent this from happening, ammonia will react with carbon dioxide in the blood, and this will produce a substance called urea. Now, this is still toxic and has to be removed from the body, but it's not as toxic and poisonous as ammonia is. Here your blood then takes the urea to the kidneys where it is excreted from the body. Now we're on to the kidneys, and this is a diagram of the kidneys. Here we can see the different parts of it. We've got the renal artery, which takes blood to the kidney to be filtered, and we've got the renal vein, which will take the filtered blood away from the kidney. Here we've got the cortex of the kidney. These parts here are the medulla, where the filtering units are found, and they filter down into this tube called the ureter, which connects the kidneys to the bladder. Now, at this point here in the kidneys, this is where excretion of urea, water, salts and glucose will happen. Now we need to look in more detail at how this filtration of the kidneys happens, and we can see here that we've got the kidney and we've taken a small section of the kidney to show what happens within this part of the kidney, which is called a nephron. Now, filtration of the blood happens in 3 stages. We've got high pressure filtration, selective reabsorption and salt and water regulation. And these 3 stages will happen at different parts of this nephron. This diagram here shows the different areas that it will happen. So this blue area here shows where ultrafiltration or high pressure filtration will happen. The green area will show where selective reabsorption occurs, and the pink or red area shows where salt and water regulation occurs. Now, as the name suggests, this high pressure filtration happens at high pressure. The blood is brought in from the renal artery and enters this vessel as it branches off, called an afferent arteriole. So arterioles are smaller branches from arteries. The afferent means to take to. Affer means to take to in Latin. Okay, to carry to. So the blood is carried to this structure here called the glomerulus. Now this is a section of blood vessel that is quite leaky. So when the high pressure blood flows through, any small soluble molecules due to the high pressure will leave the blood vessel and enter this yellow region behind the glomerulus or around it called the Bowman's capsule. Now, the other reason that this happens to get the high pressure is that this blood vessel that leaves the glomerulus is called the efferent arteriole. Effer to mean carry away. Now, this arteriole has a smaller diameter than the afferent arteriole. This leads to a bottleneck effect. To show this, I'm going to use this diagram. So here, imagine that this is going into the efferent arteriole where the diameter decreases, and what we can see is as the blood flows through, it's got less room to get through here. So what we're going to get is we're going to get a build-up of pressure behind the efferent arteriole. This will further cause more of the soluble materials to leak out of these blood vessels into the Bowman's capsule. Now, some of these molecules include glucose, amino acids, salts, hormones and water, and they are all, as much as possible, removed from the blood. This is due to the fact that it's better to remove all the waste than keep any of it. So we remove everything and then we absorb things back that we want to later, and this is what we're going to look at next. So here next we have selective reabsorption. Now this happens again in this green area here. Now in this area we've got the substances travelling along this part of the duct, and the useful substances for the body such as glucose, some water and salt are reabsorbed. Now what allows this to happen well is that we've got a folded structure for this tube to allow an increased area for reabsorption. And the last part here, which we're going to look at, salt and water regulation, this happens mainly along the loop of Henle, this loop of the duct here. Now salt and water are reabsorbed into the blood to maintain normal water levels within the blood to prevent osmosis bursting blood cells, etc., etc. The length of this loop of Henle allows more time for that normal level to be maintained properly. The loop of Henle also has a really good blood supply in order to get ideal water concentrations within the blood. So this means that if your blood is very watery, if you've got a lot of water in your blood, less water is going to be reabsorbed into the blood because we don't want it to be very watery. A lot of dilute urine will be produced. So if you've got watery blood, you'll produce a lot of urine. If your blood is not very watery, then more water is going to be reabsorbed from the kidney tubules. You're not going to want to lose that water, so you'll get a smaller volume of urine which will be quite concentrated. This process of maintaining water concentration is controlled by a hormone called antidiuretic hormone, and shortened to the name ADH. So antidiuretic means against making urine. And the way that this works is that if you have got, we want a normal water concentration in our blood. If you get too much water within the blood, that will be detected by the hypothalamus within the brain. It will trigger the pituitary gland to secrete less ADH, okay? So not as much ADH, antidiuretic against making urine. That will decrease the permeability of renal tubules, which means that we'll be able to get less water reabsorbed into the blood. This will mean that we have very dilute urine. Conversely, if you've got too little water in your blood, again detected by the hypothalamus, the pituitary gland will secrete more ADH. This will increase the permeability of renal tubules and more water will be reabsorbed into the blood, so you will get very concentrated urine. So if you're secreting more ADH, you're wanting to not make any urine, you're against making urine, so we can see that we're going to get less urine made in this situation. And when this happens, the water concentration of the blood will return to normal. This entire process is an example of negative feedback. Now, within biology, most processes will involve negative feedback mechanisms. Now, the way that this works, if something gets too high, then it's going to be brought back down to a normal level. If something gets too low, it's going to be brought up to a normal level. This is a process of homeostasis, keeping things maintained at a normal level. So it's very common to find these negative feedback mechanisms within biology. Now next we're going to look at one solution for if someone has failing kidneys that cannot filter the blood properly. So we're going to look at what the kidney dialysis machine does. So the way that dialysis works is that blood is taken from the patient and enters the dialysis fluid. It is filtered throughout the machine and is brought out as clean blood and enters the patient at a different point. This means that the patient has 2 points at which blood is removed and entered back into the body. Now, the dialysis fluid that is used is low in salt and urea, but the same concentration as the blood when regards to sugar and mineral concentrations. What makes this work so well is the fact that the dialysis machine has a partially permeable membrane. Now this doesn't allow the blood cells to pass through the dialysis membrane, however it will allow soluble materials in and out, although these must of course be small molecules. The dialysis fluid is low in salt and urea, however the sugar and mineral concentration is the same as it is in the blood. Now the reason that it's low in salt and urea is because the blood will be quite high in salt and urea. Now this will give a concentration gradient. We'll have a high concentration within the blood and a low concentration within the dialysis fluid. This means, for example, if we have lots of salt at a high concentration, it will move along the concentration gradient into the dialysis fluid by diffusion. And again, the same is true when it comes to urea. We'll have a high concentration in the blood and a low concentration in the dialysis fluid, and it will move out via the partially permeable membrane. The reason we have the same sugar and mineral concentration as in the blood is because if they are the same concentration, there is going to be no net movement of the sugar or any other substances needed by the body, and that will remain in the blood. And that concludes our look at excretion in Unit B5 Biology Revision.